Welcome to EYETALK, an interview session about issues and controversies in ophthalmology. And I'm Natalia Anисimova, and today we're speaking to Professor Petrovski from Norway. Hello, Professor.
Good morning, Natalia.
Bioengineering corneal tissue has recently gained a widespread attention. In order to facilitate corneal regeneration, a range of materials is currently being investigated. How do you think? What is real the worldwide need for artificial cornea?
Well, as we know, there's close to 8 to 10 million people in the world that have corneal blindness, and there is a huge shortage of donor corneas which are needed to transplant these people. So obviously the need is very high based on the fact that there are close to 10 million people in need of such tissue.
Have you done your clinical investigation, and how close did you approach to the clinics, or did you do only in vivo studies?
Our Center for Eye Research at the University of Oslo was the first clinic in Scandinavia actually to do ex vivo cultivation of corneal limbal stem cells. And these cells are actually being expanded for approximately 2 weeks ex vivo from the patient's other eye, and they're being transplanted to people in limbal stem cell deficiency. That's the closest project we have had where we have used clinical application of these cells. Otherwise, we have ongoing studies which are in preclinical phase, such as bioengineering of cornea and then derivatives of corneal stroma. And also we have another research line which is on producing cornea endothelial cells and sheets for transplantation.
What do you think are the obstacles for the clinical approach to integrate all these technologies into the widespread clinical application?
It's a good question because there are many differences worldwide. In Norway, as part of the European economic area, we abide to the European laws. But worldwide, actually, the major issue is to have a standardized protocol and GMP production of cells or tissues for transplantation. So the regulative, which is currently there for GMP production, is going to be a bit obstructive in how many places we'll be able to have such facilities because we know they're quite expensive to build and to maintain as well.
For GMP production, should you need a huge manufacturer or can you make it really like in a tight place so it can be portable, or if there is something interesting right now?
Indeed, as this technology has developed over time, the classical GMP rooms, probably which were found only in big pharmaceutical companies, they can be built in smaller departments. Moreover, there are several companies in the world which provide small sterile hoods, which is all in one package where you can actually cultivate the cells. It's a glove system, it's a closed system with the window system for passing over the tissue, and there is a microscope, incubator, everything is there. So actually these technologies are nowadays existing there for about 250 to 300,000 euros instead of having a huge facility. That's an alternative, maybe.
And back to discussion about the clinics, which kind of corneal pathology do you plan to treat with the artificial cornea?
Obviously, the need for cornea transplantation is high in the world, as I said in the beginning, up to 10 million people that are cornea blind. And there is a variety of pathologies, a lot of them having trauma-induced from burns, for example. But also in the future, there are other needs for cornea transplantation and cell therapy, because the dystrophies are also a huge area where it is believed that some sort of cell gene therapy will be available for treating these diseases. On top of that, we have all these chronic ulcers inflammatory disorders, and obviously a huge line of the Fuchs dystrophies and endothelial disorders of the cornea.
Recently, the traditional methods of the treatment of severe corneal pathologies are either keratoplasty or corneal prosthesis. How do you think artificial cornea, what advantages will bring in comparison to the traditional methods?
So if we analyze in the past decades, the therapy was more on the penetrating type, full penetrating keratoplasty and the keratoprothesis. But with time, these procedures have decreased, and more lamellar procedures are actually being performed worldwide. So having said that, I would think in the future, there is a great room for improvement of these lamellar procedures and use of bioengineered materials to replace either the interior or the posterior part of the cornea. I would see probably less and less of these full penetrating procedures in the future.
And what are the functional requirements or maybe anatomical requirements to artificial corneas?
Yeah, as we know, the cornea is there to protect. It's a first line of barrier for the eye, for the body, and also it needs to be transparent. So these materials which will be used in the future, they need to be transparent, but they have to have the similar type of structure and then sturdiness or robustness of the cornea. Yet at all time keeping the refractive power and the transparency which is needed there. So there are several functions which need to all be in place for artificial material to actually fit perfectly there and to be accepted by the body.
How you actually think that the cornea, actually the stromal part, in different levels it has its own biocompatibility, or it's the collagen structure is very different and it's getting more and more compatible in the deeper levels. Is it actually matters for the artificial cornea?
Anatomically speaking, if you look at the histological section of the cornea, you would see different tightness or ordering of the collagens in the anterior 1/3 to the posterior 2/3. Even our group and collaborators have shown how these collagen fibers actually behave with the hydration and different changes of the cornea. Obviously, the distribution of cells within the cornea is a bit different. So we are speaking about keratocytes and corneal stromal stem cells. Even their distribution in this structure is different. This may affect in the future the production of artificial corneas, because this in a way has to be reproduced as it would be in vivo, as compared to corneas which are probably acellularized, and those will already have the anatomical structure which should be there, and then the cells need to get in.
The main component for the artificial corneas is collagen. For a lot of for opinion of the leaders for bioengineering of the corneas. And how you think which technologies allow the collagen and artificial cornea to get more stiffer, as we know that, for example, for the penetrating keratoplasty we need to suture it and to prolong as much as we can the persistence of the artificial cornea in the eye.
Right. Well, obviously, the collagens as a structure, they can be made tighter or less tight, a bit tighter in a way as a clinical procedure of collagen cross-linking. So this is something which is done on a regular basis in different eye clinics or cornea clinics. One can also affect the tightness through some chemical ways as well, by applying certain type of, there have been tests on different types of aldehydes and alcohols and how to change the cross-linking of the collagens. That in itself having some maybe cytotoxic side effects. In long run, actually, to answer your question about the transplantation and the suturing and so on, eventually a sutureless method would be something which would be ideal. And maybe with the materials as existent today, as for example, magnetic nanoparticles and similar materials, we may be able to achieve something like that in the near future.
What are the various types of artificial corneas, and which corneas are actually more promising for the worldwide application?
At the moment, there are many attempts in different labs in the world to use different material and different origin of artificial corneas. Some groups are working on porcine acellularized corneas and porcine collagen, which can be used for spinning of these collagens into a bioscaffold. So that's using the porcine material into making artificial scaffold for the cornea. Other groups have used bovine, for example. So these are all animal materials. I must add that the porcine collagen at the moment enjoys an FDA approval for human use. And then comes the human recombinant collagen, which we know that it's very expensive to produce. There are acellularized types of corneas or corneostromas. I can envision maybe in the future the human acellularized corneas as we prepare in cornea banks. Just it will have a much longer or almost indefinite shelf life. And then comes something which is pretty exciting, I think, the 3D bioprinting of the corneas, where one can set layers of collagens in the tightness that you would like to have and also have them cellularized. Overall, the big issue in the future will lay in obtaining material which will be penetrable by the cells of the cornea. And the question will be whether we transplant these as empty ghost shells of collagens, or we would like to already embed the cells of the cornea. I would probably prefer embedding the corneal stromal stem cells. We and other groups have also shown that if they're being embedded, they can also show markers towards different directions, such as markers afterwards for keratocytes and production of de novo production of collagen, so turnover of the collagens and keratin sulfate and laminins and the like.
And do you actually use the collagen as a biometrics to embed the cells, or which type of metrics do you prefer?
Yeah, we have used in the 3D printing of the corneal stroma, for example, the matriderm, which the basis of it is skin collagen and also elastin, and that's the ground, the carrier for the bioprinted cornea. Otherwise, it's the collagen type 1, which is used in the bioprinting, and then the cells. I would prefer the laser type of bioprinting, but there are several other methods of bioprinting worldwide.
Is it xenogenic collagen actually?
This collagen that we have used is porcine collagen, but one can use also recombinant. Coming back to the issue, porcine collagen, which is relatively inexpensive, is FDA approved. So I don't see any issues of why not using it. And for many years, for example, heart valves have been made out of big material, and it has worked beautifully in cardiology.
Did you use corneal stromal cells in ex vivo experiments to see if these cells can produce the scaffolds or any kind of corneal tissues?
Yeah, we have used corneal stromal stem cells, which are mesenchymal-like stem cells. in long-term cultures where we allowed the cells to make 3D structures. And we have shown that they're capable of producing their own collagen network. And this network is, it can be seen actually on one of the slides. The collagens look similar to the normal corneal stroma. So yes, indeed, these cells, if they're allowed ex vivo to do a 3D structure, they know what they're doing. One issue with this is maintaining good transparency of these cells and having this tissue which is produced ex vivo to be robust enough to be able to transplant it. We have actually transplanted that in organ cultures of human corneas, and so far the results are promising.
Yes, I think it was a great discussion. Thank you for coming here. And it was the program EYETALK, and I'm Natalia Anisimova and Goran Petrovski. Thank you.
Thank you, Natalia. Thank you.